Good morning to all the aspirant. Today class continuation the last class. Today topic is bacteria based on flagellation. Flagella is the organ for motility. Okay, let's start. Atricus. Flagella absent. Atricus. Flagella is absent. Example: Lactobacillus. The next one is the monotrichus. Mono means single and one. Mono means single means one. Trichus means flagella. Single flagella is present at one end. That is monoflagellation. Like this picture. Okay, observe this one. This is bacteria. This flagella. One end. Okay, the next one is Leptotrichus. A group of flagella at one end. Example: spirulum. Monotypous example is Vibrio cholera. Vibrio cholerae. Leptotrichus. Group of flagella at one end. Example: spirulum. One end means Leptotrichus. Leptotrichus. This is bacteria cell. Group of flagella at one end. Like this. Okay. The next one is Amphitrichus. A single flagella at each end. Example: Nitrosomonas. Nitrosomonas. See. Bacteria at each one end. Bacteria at each end. Okay, Nitrosomonas. Example: Nitrosomonas. Okay. And next one is the Peritrichus. Flagella present all over the body. Peritrichus. This one. Flagella all over the body. This means all sides are present. Flagella. This, like this. Okay. Example: E. coli. Peritrichus. Example: E. coli bacteria. E. coli bacteria. The basing on the flagella bacteria. Then let us see structure of the bacteria. So bacteria possesses a distinct cell wall with different wall layers like capsule or slime layers plus plasma membrane, flagella and pili. With the development of the electron microscope in the 1940s, bacteria revealed themselves are more than simple spades and rods. Scientists uncovered a wealth of microscopic and submicroscopic details of bacteria and showed how very minute bacteria can be as complex as very large visible organism. Recollect the prokaryotic cell structure you have already studied in your first year. However, we will discuss two variant components of bacteria cell here. These are flagellum. See guys, I have given to the diagrammatic representation regarding the bacteria in Whittaker 5 kingdom classification in the first group of kingdom Monera. I have given to the diagrammatic representation here. Look about to the diagram. Okay. Okay, this is cell wall. Okay. The slime layer or capsule is made up of polysaccharides and amino acids and which acts as osmotic barriers. This is made up of polysaccharides and amino acids. The below layer is plasma membrane. The cell wall below is plasma membrane. Plasma membrane is tripartite in nature. Okay, This is the bacterial cell. This flagella. Mesosomes. Mesosomes are simply enfolding a plasma membrane. Mesosomes are enfolding a plasma membrane. This like this. This here mesosomes. Mesosomes. Plasma membrane contain respiratory enzyme like dehydrogenated. See, these are the plasma membrane enfolding here. See, these are the plasma membrane. Mesosomes involved in the respiration. Mesosomes involved in the respiration prokaryotic. In the respiratory enzymes. Yes. So next one is the flagella is made of flagellum. This one flagella made of flagellum. Okay, made of pili. The next one is the pili. Pili are small hair-like outgrowth present on the bacteria cell structure. Outgrowth present in the bacteria. Pili are small hair-like outgrowth present on the bacterial cell surface made of pilin protein. Pili made of pilin protein. Okay, pilin made of pilin protein. Pilin helps into information of conjugation tube and glutamation. Okay guys, the next one is based on the nature of staining bacteria. Maybe gram positive retain the blue stain or gram negative does not retain the stain. And next one is a gram staining. The next one is gram staining of bacteria. Gram staining, heat and fix it here, and after that crystal violet, rinse with water. Rinse with water. Add dilute iodine solution. All bacterial cell appears deep blue. Okay. Treat with 95% alcohol. Okay. Retail purple color, gram positive, distinct and appear red in color, gram negative. Let us see the difference between the gram positive and gram negative bacteria. See here, the main characters is the peptidoglyco layer is the main character is the peptidoglyco layer. Gram positive, gram positive, gram negative. Okay, na? Cell wall more thickness, nanometers, 25 to 30 nanometers. Gram negative bacteria or cell wall is thickness only. Thick, 10 to 15 nanometers. The next one, cell wall is homogeneous. Next one is homogeneous. Cell wall is homogeneous. This is gram negative bacteria. Cell wall is heterogeneous. Heterogeneous. Okay. Gram positive bacteria. Cell wall is more thickness. More thickness. 25 to 30 nanometers. Gram negative bacteria thickness only 10 to 15 nanometers. Cell wall is homogeneous layer. Cell wall is heterogeneous layer. Okay, heterogeneous layer. Third one is high content of the peptidoglycan, 20 to 80%. Less content of the peptidoglycan. 10 to 20. Okay, 10 to 20%. Tricolic acid absent in gram negative bacteria. Tricolic acid present. Very little lipid content, 0. Very little content, 0.2%. High lipid content, gram negative high lipid content, 20%. Only few gram positive bacteria is only few amino acids are associated with muramic acid complex. Amino acid associated with muramic complex. Large where gram negative bacteria amino acids are associated. LPS means lipopolysaccharides layer absent in gram positive bacteria, present in gram negative bacteria. Okay, this differences between the gram positive and gram negative. Okay, let us move forward. So inner to the cell wall layer there is present matrix or protoplasma, which includes nuclear plasma, episomes, ribosomes and granules. Episomes, inner layer. Okay. This is plasma. This is cell wall. Plasma membrane. Plasmid. This is plasma membrane. In the center of the bacterial cell. Is present nuclear material DNA without any nuclear membrane, naked DNA. In bacteria, double helical and circular. This incipient nucleus or primitive nucleus is named as nucleoid or genome, sometimes called single naked chromosome. And besides this nuclear DNA, there is some extra nuclear or extra chromosomal DNA which is known as plasmid. The plasmid are small circular double stranded DNA molecules that are separate from main bacterial chromosomes, main bacterial chromosome and replicate independently. This is independently replicated independently. The term plasmid was given by Lederberg. The term Lederberg given by the term plasmid was given by Lederberg. Here, 1952. Plasmids have an independent existence. Plasmids carry genes for fertility, antibiotic resistance, R factors and bacterium production. F factor of fertility factor is responsibility for transfer of genetic material. F factor. Fertility factor is responsible for transmission, genetic material transmission. R factor or resistance factor. R means resistance factor provides resistance against drugs. Then cholinogenic factor produces colicins which kill other bacteria other than which produces these colicins. The term episome is applied to extranuclear genetic material which may remain for free state, example F factor, temperature, phase, etc. The next one, according to the mode of respiration, bacteria can be aerobic or anaerobic. Each of them is true of two types of obligate and fluctuate. Obligate aerobes are bacteria which can respire only aerobically. That generally get killed under aerobic mode of respiration. Example bacillus. Facultative aerobes are bacteria which respire anaerobically under normal conditions but can respire aerobically when oxygen is available. Most of the photosynthetic bacteria that respire only. The next one is obligate anaerobes are bacteria that respire only anaerobically. They generally get killed under aerobic conditions. Clostridium botulinum. Then facultative anaerobes are bacteria which generally respire only aerobically but switch over to anaerobic mode of respiration of oxygen become deficient. See guys, in facultative anaerobial conditions they respire normally through oxygen. If their oxygen they switch on to anaerobic respiration. These conditions known as facultative anaerobic whereas in facultative they usually through anaerobically there is sufficient oxygen switch on the aerobic respiration. That is the difference between the facultative aerobic and facultative aerobic and facultative aerobic. The next one is the nutrition. The next class is the nutrition.